But I'm very happy to start the next series of talks. This is a joint talk, if I understand it correctly, between Dr. Lucie Khemtemourian from the University of Bordeaux, where she's an associate scientist at CNRS, and working on the interaction between amyloid peptides and the membrane, along with Samuel McCalpin, who's doing a good job moderating the technology today, in addition to giving a talk. He's from the University of Michigan and is a graduate student with RAMI. So I will turn it over to y'all for the effects of zinc and insulin on the aggregation of IAPP in type 2 diabetes.
All right, can you all hear me? It's the presentation, all right? Awesome. Yeah, so as Kayla said, my name is Sam. I'm a student in the RAMS lab. I'm going to act as our representative or mouthpiece here for and share some of the work that's been done by our lab. And then Lucie, our collaborator, is going to talk about some complementary stuff that she has done as well. So brief introduction. I'm sure most of this audience is pretty familiar with this at this point, especially after the past couple talks, but we're going to be focusing on IAPP or amylin, which is found in the amyloid deposits in the pancreas of individuals with type 2 diabetes. And so those fibrils are composed mainly of IAPP. So we want to understand the aggregation of this peptide in relation to the molecular mechanisms of this disease. So the most of this talk is going to be motivated by the observation that IAPP is natively packaged in these secretory granules and the beta cells of the pancreas. And within these secretory vesicles, IAPP exists at millimolar concentration for at least several hours, and there's no aggregation there at all, at least that has been seen so far. And so the driving question here is going to be, those of us who have worked with IAPP know that at this concentration, it's going to aggregate super fast in vitro. So in vivo, why does this aggregation not occur? And are there potentially naturally protective compounds or components that exist alongside the IAPP that protect from that aggregation? And so for the purpose of this talk, I'm going to be mostly focusing on zinc, but we will also touch on the insulin, calcium, lipids, and the pH effects that also might be involved in these interactions. So in the Ramamorphy lab, this work with zinc specifically was begun about 10 years ago by a guy named Jeff Brender, who I think I actually saw in the audience. So he might be pretty familiar with all of what I'm about to say. But so he started this work. Here I'm showing the THT fluorescence curves of IAPP with several concentration series of zinc. And you see at pH 7.5, the zinc shows a pretty strong inhibitory effect. As you add more zinc, the fluorescence intensity decreases pretty strongly, indicating that the amount of fibrils formed is decreased with the zinc. But at the pH, the lower pH, like what is found in the secretory granule, there's actually a much smaller effect. And zinc actually looks like it accelerates aggregation a little bit. I mean, you can tell from the lag times here that the pH is playing a much stronger role. So the zinc at this pH might not be directly interacting in a significant way, which is sort of interesting. But still at the higher pH, it is a pretty strong direct inhibitor. When IAPP is incubated with zinc for a couple days, and you take electron micrographs of what you've let stand, you see that increasing the amount of zinc incubated with the IAPP significantly reduces the density of the fibrils that are formed. But interestingly, the morphology of these fibrils is virtually unchanged. So most amyloid inhibitors will change the morphology of the fibrils, either shortening or narrowing them, or causing the formation of like amorphous aggregates instead of fibrils. But in the case of zinc, that doesn't seem to be happening. So the next question to be addressed is like, how is this inhibition occurring? And so what Jeff did was he compared the effect on the aggregation of IAPP of several divalent cations and found that the most significant effects on the aggregation are found in the cations that are known to bind to imidazole groups. This is significant because IAPP has one histidine residue right in the middle of the peptide. And so the hypothesis following this data is that histidine residue is what mediates the interaction with the zinc, causing the inhibition. And so isothermal titration calorimetry was used to measure the strength or affinity of the IAPP with zinc. Actually, an N-terminal fragment here is used, the first 19 residues, because there were issues with aggregation of the full length. But what you see is that IAPP showed a binding affinity around 1 micromolar, which is not as strong as normal metal metalloproteins, and not even as strong as some of the interactions we've seen earlier today, like A-beta and copper, but it is stronger than you would expect just for a single histidine residue. And this control peptide, which is the same length with one histidine residue, did not show the same the same heat evolution in the experiment that you saw with the IAPP fragment. And also this titration curve is consistent with coordination of 6 peptide IAPP peptide molecules per zinc. And so this is where that cartoon that Dr. Miller drew the big X through earlier, that number came from this data here, based on the zinc to peptide mole ratio. And it's certainly okay to question that. We're using a fragment here instead of the full length, so maybe some room for conversation there. But it still seems pretty clear that zinc binds to that histidine residue at the 18th position. And this is further supported by these NMR solved structures of IAPP in the presence of zinc with and without zinc in a helix promoting solvent. So this is not a physiological solvent system, but there is some evidence that that IAPP homodimers that are stabilized by helix-helix interactions are early aggregation intermediates. And so you can see the zinc disrupting that secondary structure there could potentially be acting on that early species in the aggregation pathway. And so mass spec experiments performed in collaboration with the Rudolo group at the University of Michigan showed that in the presence of zinc, this dimer formation is disrupted. So you can see in the absence of zinc, this dimer forms, but you don't see that with the zinc. The mass spec data also showed that depending on the zinc concentration, it looks like there's a second zinc binding site. So you can see as the concentration of zinc was increased, this IAPP plus 2 zinc peak showed up in the mass spec data. And so they repeated the experiments under oxidizing and reducing conditions to verify, because there's a disulfide bridge at the N-terminus of amylin, and cysteine residues are known to bind zinc, but there isn't any difference between these, or significant difference in the data performed under these conditions. So it's unlikely that those cysteine residues are involved in the second binding site. So some X-ray absorption experiments were also performed in collaboration with Jim Pennerhahn, another professor at Michigan. And so these X-ray absorption spectra were collected of the full length and that N-terminal fragment again. And so the EXAFS data in this, the wiggles in the spectrum over here past the edge, can be processed to tell you something about the zinc coordination environment. So the data, the signal noise was an issue in the full length, but in the N-terminal fragment, if you process the EXAFS data, you can come up with a distance distribution like this that you can simulate to different coordination environments to match the experimental results. And this is consistent with simultaneous coordination of 2 to 4 histidine residues per zinc molecule. The pronounced, this sort of double hump in the full length spectrum of the IAPP suggests that in the full length, it's probably closer to the 3 or 4 histidine residues per zinc. And then several NMR experiments further support the idea of some sort of oligomeric IAPP species stabilized by zinc. So probably the most convincing data here is the watching the signal depletion over time with and without zinc. So the high molecular weight species like oligomers are invisible to solution NMR because of slow tumbling. And so you see when zinc is added, the signal depletes much quicker than without zinc, suggesting that there's some large species that zinc catalyzes the formation of. Which we propose is some oligomer of IAPP. And so the interaction with zinc seems to predominate towards those high molecular weight oligomers, because you don't see much change in the remaining signal, like you would expect if you were to see the zinc binding to the peptide. So taking all that data together, we came up with this model in which, so the zinc does not directly inhibit the aggregation of IAPP in the granule. At that pH, it doesn't seem to have a greater effect, but when the contents of the granule are released from the cell, they're transiently highly concentrated in this extracellular space, which will have the higher pH. And that may be where zinc exerts its direct influence on the aggregation. And we say it does that by stabilizing prefibrillar oligomers that are off pathway. So we can argue the exact number of peptides per zinc, but it seems pretty strong evidence that something like that is forming. So at this point, I think I'm going to give it to Lucie. Lucie, are you there? Lucie?
Lucie, we can't hear you.
Muted.
We don't hear you. Lucie, you need to unmute yourself.
You're muted. Can we unmute her?
I think hosts can unmute.
We can ask to unmute.
Rams, can you unmute?
Yes, you can hear me?
Yeah.
Now, good, excellent.
Okay. I'm sorry because I don't have the hand with this computer. This is the computer of Sam and I could not do anything. Sorry. So I'm going to continue the talk about the effect of zinc on hIAPP. So as Sam told you before, we saw that zinc did have an effect on hIAPP fibril formation. And we wanted to know which part of the frequencies was where involved in the binding. So first we decide to do a mutation. Sam, can I do the next slide? I cannot.
Right.
No, wait, sorry, it's okay. So we wanted to look at the role of zinc and insulin on the residue 18 in hIAPP fibril formation. So we study by thioflavine and microscopy. We study the kinetic and the morphology of the fibrils, and we use for that 4 analogs of hIAPP, where we did 4 mutations. First one, we change the histidine by arginine to copy the mouse hIAPP. And then we did 3 other mutations, sorry, with K, E, and A to check the effect of the charge. So first, with the microscopy, we look at the fibril morphology, and this is hIAPP in the presence of zinc. So we see that we have the same type of fibrils that without zinc. And with the 4 mutated peptide, we have also the same kind of fibrils. So it means that at least zinc do not change the morphology of the fibrils. And then when we look at the fibril, the kinetics of hIAPP fibril formation, in black, this is the fibril formation of hIAPP alone. And then in the 4 different colors, we have 4 different concentrations of zinc. And we can see that zinc slows down the kinetics of hIAPP fibril formation. If we look here at the maximum THT intensity, we see that without zinc, we have the highest plateau. So with zinc, we have each time a lower plateau. And the TNALF is increase a bit at the lower ratio of zinc. Then we did the same experiments with the 4 mutated peptide, so with arginine, lysines, E and A. And we see that for 2 of these peptides, there is almost no change in the kinetic of fibril formation, while for the other 2, we have slightly changes in the kinetics. So we study by NMR this binding. So we see that the zinc could bind to the histidine protons by NMR, and if we do integrate the signal over time of you can see that you can follow the monomer depletion over time. And we can see that for the wild type, for hIAPP, we have a decrease in the signal, suggesting that by telling us that we don't have any monomer after 1 day, and most of the species are in oligomer, huge oligomer or fibrils. If we add zinc to the same sample, we also see that we have a decrease in the integral, in the signal. suggesting also that we have we have no monomer. However, for this mutated peptide, we also see a decrease, but it's less important. We still have 50% of the signal after 1 day, suggesting that we still have monomer after 1 day of experiment. But with this peptide and with zinc, then we see a much more important decrease, suggesting that this zinc also interact with other amino acids than histidine. So we in collaboration with Birgit Strodel in Lucie Institute, we saw that the hIAPP could bind to the membrane with the N-terminal part. So the N-terminal part here, we can see here, is next to the B layer. So this is the level of the B layer. So the N-terminal part is involving in the membrane interaction. So we did the same experiments as previously, but in the presence of membrane. And then we see that the zinc does not affect the kinetic of fibril formation at all. Sorry. In the presence of membranes for human IAPP. And then we did the same experiments. Sorry. So then we have an hypothesis that zinc also binds to the N-terminal part of IAPP, because when the N-terminal part is involved in the membrane interaction, there is no change in the kinetic of fibril formation. So we did the same experiments with the mutated peptide, and for 2 of the peptides, there is no change at all in the kinetic of fibril formation. However, for the 2 mutated peptides R and K, we have an increase in the lag time, but only for a really high, zinc IAPP ratio. So finally, we wanted to know, since in the beta cells of the pancreas, where IAPP is stored, as Sam just told you, we have not only zinc, but we have insulin, and the pH is acid. So we wanted to know if insulin and zinc together could have an effect on IAPP fibril formation. So first, we study the influence of insulin alone, only insulin, on IAPP fibril formation. And as expected, we see an increase in the lag time when we had insulin. And on the microscopy, we can see a nice fibril for IAPP. And when there is fibril presence, we can see insulin presence, sorry. We can see some fibrils here, but we can see also aggregate. It could be small or bigger aggregates. So insulin does inhibit IAPP fibril formation. And recently, we did some experiments. My student did some experiments. So sorry for the slide, but I just did a snapshot of our results. So to mimic the secretory granules, we work for these experiments with a pH at 5.5, and we add IAPP with zinc and insulin. And on the top, this is where there is no insulin. So there is blue, this is IAPP without insulin. And in green, you can see that there is a zinc doesn't have so much effect on this pH, at this pH on IAPP. And when we add insulin, we start to have an increase of lag time here. And when we have insulin plus zinc, we have the increase is even bigger. So we have a synergic effect of insulin and zinc. And sorry, and if you look at the fibril morphology, so this is IAPP alone, you see only fibrils. Here with insulin, so as I told you before, you see amorphous aggregate. IAPP plus zinc, you still see fibrils. However, IAPP plus insulin plus zinc, you do not see fibril anymore. You see this kind of small aggregate. So, So now I will let Sam finish the talk.
Yeah. So so where our heads have been at recently is to look at how, as you build up the cellular environment, how different factors might interact to cause an effect on the IAPP aggregation that's more than just the sum of their parts. And so there's this evidence that this also is relevant to exogenous inhibitor molecules like EGCG. And so in the future, we are hoping to investigate how the different factors work together to affect the IAPP aggregation and how we should consider that when designing the inhibitor molecules. And with that, I'll quickly acknowledge all the previous students and postdocs in the Khemtemourian lab who actually did all this work, and our collaborators as well. And Lucie, if you want to acknowledge your students as well.
Yeah, I would like to thank my student who work on the project, and Birgit who did some simulation, and her student, of course. Thank you.
Thank you both for a great talk. Really nice job working together to get that pulled off. So I think we have some questions in the chat from Nicholas Fuller. Why would we expect there to be a lower plateau with zinc?
Well, I didn't
Plateau of what?
Sam?
Plateau. You're talking about, if you're talking about with the THT data, the lower.
I think so, but I didn't expect a lower plateau with zinc. Well, I do see a lower plateau, but I didn't expect it. I first thought that, well, the intensity of the plateau, it's quite difficult to argue, but if we correlate the intensity of the plateau with the morphology of the fibrils and you see on the microscopy, then you can kind of conclude of something. So I see a lower plateau, and first I was thinking maybe I have less fibrils, or the morphology of the fibrils are different, and the morphology are the same. So it's probably due to less fibrils.
Thank you. We have another series of questions. Let's get through what we can in the time we have left. From pH dependency on zinc effect on IAPP aggregation, can you derive a pKa? Could it indicate involvement of another ionizable group?
Yeah, that's an interesting question. I've never thought of that. I'd have to think about that more to confidently give an answer, but yeah, I think that's an interesting idea.